Okay, hello everyone, and welcome to the second of our non-COVID webinar series, today titled Proteomics Beyond the Horizon. This is the 10th webinar overall this year. Today we're happy to announce that we're joined by Professor Catherine Lilly, who's our guest chair for this webinar. Catherine Lilly is a professor in the Department of Biochemistry and a member of the Milner Therapeutics Institute. She's also director of the Cambridge Centre for Proteomics and a director of studies at Jesus College. The Lilly Group focuses on the development and application of technologies to measure the dynamic proteome in space and time in cells in a high throughput manner. And more specifically, they're interested in looking at the changes in abundance, location, interaction partners, PTM status, and the structure of proteins during dynamic cellular processes such as differentiation. So she's going to be a great person for chairing today's webinar. I just have a few of the usual housekeeping points to go over before handing over to Catherine, who will then introduce today's speakers and chair the question and answer session at the end of each talk. So as always, we're using our Slack channel for questions and discussions. So please join us there to ask questions, share any thoughts and discuss the work. The Slack really helps us to prioritize questions that others want to hear the answers to. So use the thumbs up to let us know those questions you'd like to hear answered. Slack also allows the speakers to answer any other questions or follow up after the talks are finished. We'd really like to update our website pages after today's talk, and we'd ask everyone who's joining today to maybe send a selfie or a screenshot of themselves watching the webinar to the Slack channel or the email address that we're going to show on the final slide today, so that we can create some new images on our media pages. We've had over 200 registrations this week, so if you could tell us where you're watching, that would be absolutely great. And for those needing an attendance certificate for this webinar, the details will be available on how to get this after the last slide. So once again, we'd like to say a big thank you to the European Proteomics Association, the British Society for Proteome Research, the Young Proteomics Investigators Club, the London Proteomics Discussion Group Committee, and for all their help in setting up this webinar. And thanks also to the London Biological Mass Spectrometry Discussion Group, the London Metabolomics Network, and the News in Proteomes research blog for promoting this event. We're also really grateful for Imperial College London for providing us with webinar support. And thank you to the speakers and the guest chairs for their time today. We're really pleased to announce our next webinar will be on Friday the 21st of August, 2 p.m. British Summer Time, where we will revisit the COVID-related talks, and we'll be hearing from two new speakers, Dr. Miroslav Sander and Dr. Rod Chalk, on their work in this area. So now I'm going to hand over to Catherine, who will introduce the first of today's speakers. So it's over to you, Catherine.
Thank you very much, Jo. So it gives me great pleasure to be the guest chair today, and I think there's a phenomenal lineup of speakers. So without further ado, I will introduce the first one. So the first speaker today is Dr. Ben Neely. So Ben's beaming in to this call from a long way away from Charleston in South Carolina. He's part of the National Institute for Standards and Technology. He's got a very diverse background from microbiology, wildlife disease, cancer, biomarker discovery, and bioinformatics. And he's currently focused on large-scale generation of standardized proteomics data, focusing on data-independent acquisition methods across non-model organisms. So inspired by the current coronavirus pandemic, I suspect, he's going to be talking to us today about exploring the protein landscape in non-model systems, insights into potential host virus interactions and disease resistance. So thanks very much, Ben. It must be breakfast time for you in Charleston. So take it away.
Thank you, everyone. Thank you for that introduction. And I want to thank the organizers. I've thoroughly enjoyed watching this series. It's been probably the best part of the pandemic, is getting to come from 6,500 kilometers away and get to watch. And there's going to be a lot of people that I'm going to be talking about in this talk. But again, I'm going to be talking about comparative proteomics, kind of why I love it, a little bit of how, but a lot about why you should be doing it. The last few months have been really just amazing lectures in this group, specifically about the pandemic, right? So we've had people talking about glycosylation of the spike protein. We've looked at the receptor binding domain, how it interacts with ACE2. There have been amazing talks about viral detection, protein-protein interaction analysis, biomarkers, and host response. And what's been amazing about this is how quickly all of this happened, right? I mean, these talks really, at best, maybe you were starting in March or in February. And I think that just really emphasizes how quickly science can move and how amazing, really, I think a lot of people are on this call right now. This is some soon to be released data from Magnus Palmblad, where prior to the pandemic, unsurprisingly, there was a lot of research going on, about risk and host, related to virus. But once that happened, and this is not surprising at all, is you had this enormous explosion, right? I mean, we all know there's a lot of publications right now on COVID, and a lot of that is focused mostly on the virus, right? And kind of what I want to talk about today is that, yes, this is a novel virus to humans, but it's not necessarily novel to other mammals. And what do I mean by that? So, we've had other, virus spillover events happen all the time. And essentially what's happening, and need to go ahead and say this, I'm not a virologist, but I get to hang out with people that are, so if I get something wrong, I'm sorry. I'll try to correct it. So you've got these natural hosts, right? And they're going to be kind of this transfer that goes to an intermediate host, which we may or may not know. So, in the original, 2003 SARS epidemic, that probably came from a palm civet. You've got the camels and MERS, and we know these things, right? And they eventually go to humans, and that's where we get this happening. But instead of talking about the humans, what I think is something that maybe deserves a little bit more attention are these natural hosts, right? And it's not just bats and coronavirus, right? Now, I don't want to talk a lot about bats right now because, yes, bats do have lots of viruses, but there's other things that have viruses as well, right? Cute little deer mouse, this is the hantavirus. More locally here in Charleston, we have raccoons and we have a lot of rabies cases. It's somewhat surprising. And the point here is just that there are many natural hosts for a lot of different viruses. And part of the point of this talk is to try to make people, I think, excited to do this research. And you might be thinking, well, I don't have these samples sitting around. I would want to encourage you to probably reach out. There's more than likely an enthusiastic wildlife biologist sitting around with samples and they know nothing about proteomics. One example of that, again, locally here in North America is there's a ton of research with white nose syndrome across many bat species. And this is something that I think is a very easy entry point for proteomic analysis. And you're probably thinking to yourself, well, how do I study all these species? I need databases, right? This is always the problem. Real fast, how do you get protein databases? There's genome sequencing, and then there's actually genome assembly. And this is something that in the last 5 years has gone off the rails, right? I mean, it's in a good way. We've got genomes dropping almost every day. Right now, I think we're up to about 10% of mammals, so it's called 500. But the next year, the next few years, that's really going to accelerate. And then you have to annotate, right? And this is really where the current bottleneck is. So of those, maybe 500, you don't actually have that many annotations. But real fast, where do you get your annotations? I think NCBI RefSeq does an amazing job. Right, I checked yesterday, there's 144 mammal species with annotations. So that doesn't mean that's how many there are, that's how many species have them. There's a group called DNA Zoo. They have about 109 in-house mammal annotations. I think it's 42 on Ensemble. I'm not quite sure about Uniprot. But the point here, I'm not really getting into this technical details of the talk today, but you can bug me later. But the point is that don't be put off just because there's not a database for the species that you want to work in. And we can talk more about that offline. But back to our bats, right? I mean, turns out there's a ton of bats, right? I mean, they're about 20% of all mammal species, yet you only have about 50 genomes are sequenced with only about 10 species annotated. But that doesn't mean that we can't do that research, right? Again, I'm going to kind of leave that there and go back to kind of big picture about why we're doing this. So again, we saw all these amazing things that we have done with the SARS-CoV-2. And I think part of that is, well, could you also do these similar type of studies in natural host? And really what I'm trying to get at is this whole immunology, right? It's an excellent piece was written a couple days ago. And part of this is because infection is not virulence, right? Just because it can infect the receptor doesn't mean it's virulent. And virulence is complicated, and it's basically linked to the immune system. And the start of that is this innate immune system. And for us, and really where all of this began, is what do natural hosts even look like? Like at the molecular level, what do they look like? We can say, oh, well, based on genes, we think it looks like something. But what do they actually look like? And this is where we get into this idea of molecular cartography. So in other words, the genes can tell me that roads are there, but proteomics is going to tell me how the roads are laid out, right? You know, what are the abundance of proteins? How are they interacting with each other? And I think this is the strength of proteomics. So I'm going to give an example of a study that we actually had started a long time before this pandemic. We had vampire bat serum from 17 bats from Central America. And I'm going to give some technical details here, but not go into them. But essentially, it was non-depleted, tryptodigest, short gradients, DIA. We used direct DIA analysis, and something that, again, I'm not going to go into much, but it's really cool, is Phil Wilmarth has a tool that essentially can take my protein IDs that are, you know, vampire bat and can turn them into human orthologs, and that helps a lot with our not just downstream analysis, but also comparing. From this analysis, though, you know, what does vampire bat serum look like? It turns out it looks like mammal serum, right? So you've got this huge dynamic range, is what I'm showing on the slide here. So, you know, we've got some proteins that account for most of the blood, and then it kind of drops off, just a very normal looking non-depleted serum. So what do the top 20 proteins look like, right? And again, I'm not going to go into this because honestly, this looks pretty normal, right? There's nothing that's striking in these top 20 proteins. It looks like most mammal serum. We also want to look at, well, what happens if we look at just proteins involved with complement activation? And not to go through this whole list, but again, I don't think this looks incredibly abnormal. There are some standouts. For instance, you know, why does complement C6 and C8 seem so high? It's hard to make that call. Also, something that's come up in other talks, especially Max Crispin's talk about, you know, the mannose binding lectins, right? There are mannose binding lectins in human serum and vampire bat. So I'm not giving you, you know, the answer to life, but I think what I am trying to demonstrate here is that we don't know what these animals look like, right? We don't know what it, on a very foundational level, like what's your first question? What does the blood look like? And I think you have to answer that first before you can move on. And, but bats are also important beyond viruses, right? So beyond, you know, before SARS-CoV-2, we were still looking at bats. And one of the more popular reasons is longevity. Bats shouldn't live as long as they do for their body size. This is the Peto's paradox, right? But there's also a lot of reasons to look at other creatures, right? This is an amazing figure from Peter Stinwinkel, where, you know, there's a lot of different animals studied for a lot of different reasons. People study cancer in elephants, people study hibernation in tenrecs, you know, the killifish. There's all sorts of cool things that people are studying because that organism's phenotype is somehow related to a chronic human disease. For us specifically in our lab, the thing that kind of got us started down this road was looking at diving mammals. So just real quick on what are diving mammals. You know, diving mammals are, you know, they can dive thousands of feet for over an hour. When they do that, they decrease blood flow to some organs, and when they surface, they take a big breath, they reperfuse with oxygen-rich blood. If a human did this, this would be a heart attack, a stroke, or acute kidney injury. Yet they do it, you know, countless times a day. And that's kind of strange. And so one way to ask the question of why are they able to do that is compared to some of their, you know, closely related species. So for pinnipeds, let's compare it to dogs. For cetaceans, let's compare it to things like bison. Neither of these animals dive. So how do we do that? Well, and this is kind of where I get into my second point today, is if we want to study these big questions, we need to look beyond, you know, just diving mammals versus humans. We need to look across these clades. And this is a project that Mike Janik at College of Charleston and I began, which is the Compare program. And essentially, we're just trying to profile as many proteomes as possible, focus on mammals initially, and all the roadblocks that need to be overcome to make that happen. So again, for this first phase, we had serum from a lot of different zoos and wildlife biologists. Really big shout out to those people. And these species were distributed across Aphrotheria, even-toed ungulates, carnivora, odd-toed ungulates, chiroptera, primates, and the guinea pigs, mice, and rats and rabbits. What we did was a pretty vanilla approach, a non-depleted serum. This was DDA, just normal searching. And again, we're actually converting everything to its human ortholog so that we can compare it, right? Same thing that we talked about before. So I'm going to give a story that is not a success story, unfortunately. But the first question was, well, what if we do this initial question of diving, right? Diving versus humans. So I know that the cetaceans share 131 proteins. I know that the pinnipeds share 158 proteins. If I compare that to my humans, I know that, not surprisingly, there's quite a bit of shared proteins. I mean, mammals are mammals. They on some level look very similar. But there was this list of 7 proteins that were unique to our diving mammals. And one of them that really caught my eye was this peroxyredoxin 2, sorry. Because we're like, oh man, this is a redox protein. Maybe this explains everything. It's at least under, it is present in humans, but it's not as high as these diving mammals. We thought, oh, this is great. But again, we collect all this data, right? So let's look at peroxyredoxin across the whole tree of life. And what you can see is there's not a pattern, right? Like it's not just diving mammals. It turns out the buffalo, yeah, he's got it. The dog, he's got it. It is missing in humans and it is missing in these other random species, but is this the answer to the diving phenotype? Maybe not. But it really emphasizes that you need to do these bigger comparisons. And to keep it short, I think I'm still good on time, who knows? In summary, I think it's incredibly important to get into this molecular profiling in species that we don't normally think about. I think technically it is possible, and I think that's what we've demonstrated, but proteomics is also very unique, especially with biofluids, because you can't sequence a erucic bat genome and know what the proteome of the blood is. Right, that's just not how genomics translates, right? It's made in the liver, it's secreted into blood. There's a lot going on there. And again, like I said before, I think there's a lot of mass spectrometry ready projects out there. Just ask, talk to a wildlife biologist. I know people, I'm sure other people know people. These aren't samples that you need to go out and get. These are probably already there and they're probably pretty cool. More about the Compare project, we have demonstrated that we can generate and compare proteomic data from multiple mammal species. I think what I was trying to demonstrate here is that you need to do more than A versus B, buffalo versus human, and mostly because you need to limit these false positives. And so these clade level comparisons are essential. And something that I think we're starting to see is that maybe wild animals just look a certain way. We see certain things that we think are special, and then we look across and we're like, oh, well, it turns out all wild animals have this, which kind of gets into this other point is, maybe it's not that wild animals are special, maybe it's that humans are just kind of weird. And if we don't know this, and part of that is just because we haven't really looked. And so that's really where I hope everyone is somewhat motivated to go and do this kind of work. I want to give a big shout out to the College of Charleston, Mike and Allison, Magnus, Phil, and Daniel Becker. And with that, can take some questions if we have time.
Great, thank you very much, Ben. That was extremely thought-provoking and something that's a bit different, as in the organisms that you're studying. We've a couple of questions, and I've got some questions as well. So here goes. This is from Harry. What's the most appropriate biological source to study proteomics in the context of viral interactions? Serum, lung tissue, for example?
Yeah, so this is the problem with wild animals, right? Is you have like what you would like to have, right? It would be amazing if you could create cell lines from from a bat's lung tissue or from other parts of the bat. But what you could actually get in a real situation is probably blood. I mean, this is why we do a lot of work in blood. Blood's horrible, but there's people with huge stacks of blood spots, and we know that we can do proteomics on blood spots. So is it perfect? No. Will it at least give us a clue about do they have crazy levels of complement? Do they have weird levels of other, proteasomes, guanylate binding proteins? So perfect world, I think generating cell lines from some of these species would be amazing. That's probably not going to happen very easily, but you can today do blood samples.
Okay, thank you. A one now from Claire. You looked at the redox status of proteins in the serum of diving versus non-diving animals. Is this true? Were you looking at the redox status of the proteins, comparing diving versus non-diving mammals?
We didn't at all, right? So this was a pretty basic, kind of protein profiling experiment. I mean, if that's something, I'm not even exactly sure how I could set that search up, but we have that data, right? If that's something that we can get from that data, I think that should be used. And something I did note is these samples aren't necessarily collected while they're diving, right? And that is kind of a big caveat, right? These aren't like the beluga did dive and then we took a sample. So, is that going to be reflected in the protein data that we have? I doubt it, but it could be.
Okay, another one. Can you comment on the possibility of making a prediction system for new viral threats by profiling the proteomes of wild animals?
Yeah, I think this is actually a really great point. So there's been a lot of good work looking at zoonotic potential and spillover based on, genomic work, right? So just looking at homology. So if a host, if I look like a bat, that means the bat can give me things, or these intermediate hosts. I think we can use proteomics and especially mass spec to get a better handle on, there's, 1,400 bat species. We know that they're natural hosts. I think if we can better understand them and we can better understand what viruses they may have that cause the biggest threat to humans, I think that's a very straightforward answer, that's a very good approach, is what I'm trying to say. So yeah, I think there's huge potential there.
Okay, great. There's another one just come in. Can infected bat blood be analysed for antibodies against COVID-19?
I don't see why not, and that's something that I kind of left out. These vampire bat samples, for instance, were just taken from bats for a totally different reason. We know that they have certain viruses that can infect them. We just using the data because it's DIA, right? We should be able to mine deep. We actually found a viral protein related to herpes virus, right? Which we know that they should have herpes virus infections. So I think it's not just can we find antibodies to these viruses. I think we should be definitely with DIA, and especially if you went after virus proteins, I think you should be able to detect these things in bat serum, no problem.
Great, and if I could just finish by asking a question about the Compare program that you've got going. So what tissue samples are you comparing across the different animals, and also are you taking animals that are living in very extreme conditions, such as animals in Antarctica that perhaps evolutionarily speaking have been cut off for many years?
Yeah, no, that's a great question, and that's kind of the exciting. Like when I say exploring, I literally do mean that. We're like molecular explorers. We're using serum because that's something that we can get. Most people have serum, so they've taken serum for some other type of test and they have it banked. Yeah, it would be amazing if you had a collection of livers, let's say, or muscle tissue. We don't have that. As far as other species, yeah, we've got blood from belugas. We've got blood from Weddell seal. We have if it's a mammal and we know someone working on it, we try to get that. We have narwhal, right? So narwhal's in there, narwhal, beluga. Whales are pretty hard to get because they typically don't get sampled. But in a perfect world, I think analyzing those tissues would be amazing. There's some great comparative work that's been done in other species looking at those tissues themselves. But again, we can get serum from hundreds of different species today. It's a little harder to get liver from more than like 10, right? So.
Okay, I think, Ben, thank you very much. It's a great talk, really clear, very interesting stuff. There's been more questions that have come in. So can I suggest that we take those offline and you reply using the Slack channel. So thanks very much, Ben. You can go and get your breakfast now.
Thank you.
So our next speaker is Claire Eyers. Claire is Professor of Biological Mass Spectrometry in the Institute of Systems and Molecular Integrative Biology at the University of Liverpool, where she's also the Director of the Centre for Proteome Research and Deputy AVPVC for the Faculty of Health in Life Sciences. She's also, very excitingly, just been elected as a Fellow of the Royal Society of Chemistry. So congratulations, Claire, very well deserved. She is the new British Society for Proteome Research Lecturer. Each year, BSPR sponsors a lecture, and I believe this is your first outing in this capacity. So tell me if not. And she, I understand, because I've been BSPR lecturer myself, that she can be invited to all sorts of different conferences, departmental seminars up and down the country, and BSPR will support her in that. So if any of you in your institutions would like to book Claire, then she is open for bookings, I believe. So her research really centres around understanding the relevance of post-translational modifications and their roles in regulating cellular signalling in health and disease. She has expertise in development of MS-based methods using all sorts of different separations technologies, including eye mobility separations. She's pioneered how to capture non-canonical phosphorylation on amino acids that most of us don't really think of as major targets for kinases. And that's because they've not been very easy to analyse. But Claire has shown extraordinarily impressive perseverance in trying to detect and coming up with methods to detect these non-canonical phosphorylation events. And her perseverance has won out because she's now got really cool technology, which I'm sure she's going to explain to us at her fingertips, which she's using to gain fascinating insight into the role of these non-canonical phosphorylations in all sorts of different cellular mechanisms. So they're underpinning a wide variety of different cellular mechanisms. So Claire, I've not heard you speak on this subject for a couple of years now, and I'm really looking forward to this. Title of Claire's talk is Expanding the Landscape of Human Protein Phosphorylation. So Claire, the floor is yours.
Thanks very much, Catherine, although I think you've given away half of my talk, but and thanks very much to the organisers. This is my first virtual seminar, so let's see how we get on. So as Catherine said, I'm going to be talking about phosphorylation today. If I can change the slides. I've been working in phosphorylation really for the whole of my career, so about 20 so years. But I appreciate that many people might not be quite as familiar with the protein phosphorylation field as I am. So just as a little introduction, because this is going to become quite important later. Protein phosphorylation is catalyzed by protein kinases, and they use the gamma phosphoryl group of ATP and then transfer that to, generally speaking, serine, threonine or tyrosine residues, particularly in mammalian cells, where the vast majority of phosphate goes on to serine residues. And the addition of that phosphate will change the catalytic activity of proteins. It can also change binding interactions, it can change their stability, and can also change their subcellular localization. And because of these fundamental effects on the protein and protein function, protein phosphorylation has a wide range of effects on different cellular processes. So it does regulate cellular proliferation, differentiation. It's critical in regulating gene expression, apoptosis, immune response, inflammation. You can see the list here, short, long-term memory, and a whole variety of other things. But because phosphorylation is so critically important in what the vast majority of our cells do, you see abnormal phosphorylation in a whole range of diseases, and also you see this as both cause and consequence. So disease can cause abnormal phosphorylation, and abnormal phosphorylation can cause disease. So because of this, protein kinases, so the enzymes that regulate protein phosphorylation, are the critical target in most of pharmaceutical industry. So it's one of their most major drug targets. So the way this generally works is you will identify a protein kinase which is upregulated or differentially active in some form of pathological condition, and they will design a small molecule which inhibits the activity of that protein kinase and can induce remission under the right circumstances. Roughly 30% of R&D spend in pharma is on development of protein kinase inhibitors, and this amounts currently to about $20 billion per annum, and this is growing quite rapidly. At the moment, there are 71 FDA-approved kinase inhibitors, largely for cancer indications, but also non-cancer, such as arthritis, for example. And there's 100 plus
kinase inhibitors that are currently in late stage clinical development. And all of these, or almost all of these, small molecule inhibitors target this ATP binding pocket that you see here. So protein phosphorylation and protein kinases, so the enzyme that regulate protein phosphorylation, are important not only to understand actually what is going on in cells, but also in terms of developing treatment and therapy. So I mentioned about serine, threonine, and tyrosine phosphorylation in mammals, but plants, fungi, prokaryotes, they use different phosphorylation mechanisms. So they work predominantly using the two-component signaling system that involves phosphorylation of a histidine residue here, and then that histidine phosphate group then gets transferred to an aspartate residue on a receiver domain of an effector molecule, and it's that which induces a cellular response. Now, there's no known functional homologues of these two-component signaling system in vertebrates, so for years it was thought that they weren't really relevant. So serine, threonine, and tyrosine phosphorylation was really the major mechanism of phosphorylation-mediated signaling. But there's been evidence, probably over the last about 15, 18 years, which suggests that that may not necessarily be true. So there is elevated histidine kinase and phosphatase activity, for example, in certain carcinomas, so hepatocellular carcinoma. And more recently, Tony Hunter's group has managed to make antibodies against phosphohistidine, and they have shown that using these antibodies that histidine phosphorylation can regulate ion channel regulation. It also appears to regulate certain DNA and RNA binding processes. And it's possible actually that the switch from a positively charged histidine residue to a negatively charged phosphorylated histidine actually may regulate binding of proteins to nucleic acid purely through an electrostatic interaction switch. One of the really interesting things about the antibodies that they generated, and they generated antibodies against both the 1 and the 3 phosphohistidine isomers, is that actually if you look at the subcellular localization, they're quite different. So 1 phosphohistidine, for example, seems to be everywhere but the phagosomes, which is an acidic compartment, and then 3 phosphohistidine, at least in these particular experiments, stained to the centrosomes and the spindle poles. So there may even be different roles for these 2 phosphohistidine isomers. Okay. Now, anybody who has done or heard about phosphoproteomics will know that the vast majority of phosphoproteomics studies will employ some form of phosphopeptide enrichment, so that from the complex cell lysate that you start with, you can enrich your negatively charged phosphopeptides using iMac or titanium dioxide, and then it's these that you separate by reverse phase LC before you do your tandem mass spectrometry. And for serine, threonine, and tyrosine, the phosphomonoesters, these are stable at low pH, and this works really well. But for phosphohistidine, this is no longer a phosphomonoester, this is a phosphoamidate bond, and this is acid labile. So the time it takes and the pH at which these types of phosphopeptide enrichment strategies are formed, it means that you lose that histidine phosphate. So the standard workflows are not actually suitable for acid labile phosphohistidine. And so for about the last 10 years, probably about 10 years now, we have been trying to address this and say, how can you enrich for phosphorhistidine containing peptides to identify sites? Because based on the antibodies, they appear to be, phosphohistidine appear to be quite prevalent in mammalian systems. Okay, so titanium dioxide in iMac is not actually the only way of enriching for phosphopeptides. You can use anion exchange, as this really nice paper by Andy Alpert and Carl Mettler showed. And this works in a similar way to iMac-based studies, in that you are using the electrostatic interaction that you get from your negatively charged phosphorylated residue, and it is binding to your anion exchange. So phosphopeptides will have stronger interactions than non-phosphorylated peptides to anion exchange, and the same is also true if you have a phosphorylated histidine residue. Now, like most studies which fractionate samples pre-going into the front of a mass spectrometer, you don't generally want to increase the amount of salt you had. So the way that they were doing this analysis at the time was by changing the pH and therefore decreasing that electrostatic interaction. We didn't want to play around with pH because obviously, as I said, acidic pH causes us problems. So we modified this SACS protocol to actually use a volatile salt, which then is relatively easy to clean up. So just in brief, then we generated some phosphohistidine containing peptide standards. You can't do this using standard FMOC based synthetic chemistry, but you can do this in vitro using potassium phosphoramidate. So we took a standard protein myoglobin that we got out of a Sigma bottle, treated this in vitro with potassium phosphoramidate, and you can see here a phosphorylation pattern from intact mass. So we've got at least 5 phosphate groups on this protein. And then using a combination of collision-induced dissociation, and this actually is electron transfer dissociation, we can map the sites of histidine phosphorylation on these peptides. So using these standards and a combination of serine, threonine, phosphorylated alpha-beta-casein standards, we then subjected these to strong anion exchange using a modified buffer system where the pH was stable at pH 6.8. And what I'm showing here is pooled fractions across this strong anion exchange gradient, where the grey is quantified non-phosphorylated peptides and the blue is quantified phosphorylated peptides. And you can see that the vast majority of phosphorylated peptides come out in the latter fractions because they have these stronger interactions with the solid phase material. So we are separating large proportions of the phosphorylated peptides away from the non-phosphorylated portion. And this is how we managed to retain histidine phosphorylation. Okay, so we have a method for separating histidine-containing phosphopeptides. So what did we do with it? Well, the first thing to do was actually to take HeLa cells and to see how many phosphohistidine-containing peptides we could identify. So we ran through this protocol. We lysed cells. This is actually a urea-based lysis buffer so that we could maintain pH, separated it by strong anion exchange fractionation using the approach that I outlined briefly before. This is at pH 6.8, and then used a multi-mode fragmentation strategy where peptides were subjected to higher collision-induced dissociation, where neutral loss of phosphate, so 80 or 98, then triggers EPHGD. And then those data were analysed using MASCOT for peptide identity and PTMRS to understand site localization confidence. And what we saw, there we go, is that if you look on the right hand side here, that the number of phosphohistidine containing peptides, either total peptides or unique phosphorylation sites, was actually broadly speaking to the number of phosphotyrosine sites and peptides that we identified under the same conditions. Slightly lower, and this is irrespective of whether or not we are using the standard class 1 phosphosite localization or whether we increase the site localization confidence by increasing the PTMRS cutoff score. And you can see an example of one of these phosphohistidine containing peptides here. Even though that histidine is adjacent to an SPTP motif, based on the fragment ions that we see around here, we have high confidence about histidine phosphorylation. Using the high confidence sites that we identified at 0.99 PTMRS score, we can start to look at sequence conservation. And what's really interesting is that we can see a preference for certain hydrophobic residues. So leucine, for example, isoleucine and valine at the +1 position relative to the site of histidine phosphorylation. So it's starting to give an indication that there may be some motif-based preference for phosphorylation of histidine. So as Catherine mentioned in the introduction, it's kind of given away with the big reveal, it's not just histidine that is phosphorylated. There are a whole load of other residues, amino acids, that can be phosphorylated. And having developed the methods for histidine, we realised actually, and this was fairly late on, I would admit, that the methods that we developed would actually be suitable for looking at all these other types of non-canonical phosphorylation. So while you see 2 isomers of phosphohistidine, you can also see phosphorylation of lysine and arginine here. You can also get phosphorylation of aspartate, which we knew already actually from the 2 component systems, but you can also get phosphorylation of glutamate and phosphorylation of cysteine. So if we search the data using all of these types of non-canonical phosphorylation residues, we actually start ending up in a little bit of a quandary. And this is to do with the way that we generally do the bioinformatics searching. So if we consider this as a theoretical problem, when we consider one type of modification but 3 potential sites, we are then asking our database search engine to consider 8 possible states that we can see that peptide in. If we increase this number of potentially phosphorylatable residues to 5, then we end up with 32 possible states for consideration by the search engine. By the time we increase that to 9 possible phosphorylation sites, then we end up with 512 possible states that we need to consider, and that is a massive increase in search space. Another problem that we have is also about statistical confidence of the identification, because as you increase the number of potential variable modifications, then the statistical confidence you have of the identification decreases quite dramatically, and I'll go into this a little bit more later. But one of the ways that we went around this problem of increased search space was actually by decreasing the size of our database. So we were generating in the region of 750,000 spectra per bioreplicate for this experiment. And this is just not really tenable to then search the whole of the human proteome, which has got some, 20,000 sequences. So what we did was actually generate a new database that only comprised those proteins from the cell lysate that we had actually identified. Now, bearing in mind that the SAX strategy, the UPAC strategy that we developed, it identifies and it separates both phosphorylated and non-phosphorylated peptides. It's just that you are basically sequestering a significant portion of your phosphorylated peptides to those later SAX fractions. But what it means is that you are still getting quite decent coverage of the total proteome in the early stage of that fractionation round. So reduce the database size, so we went from 20-something thousand sequences to just over 7,000, and reducing the database complexity therefore threefold, and that had quite a big difference to the way that the search is run. So using this strategy, we then searched for all of these non-canonical residues. So I'm showing you here only at PTMRS 0.9. You've seen this data before, so this is looking at histidine compared to tyrosine. But what we see for the other types of non-canonical phosphorylation is actually significantly more phosphorylation sites for aspartate, glutamate, the lysine and arginine. You'll see 2 sets of numbers here, which I'll talk about in just a moment. And we also see around 70 phosphopeptides that containing cysteine. This is mapped here then according to site localization confidence, where red is everything identified greater than 0.75, and green is everything that is greater than or equal to 0.99. So the really high confidence, what we think are high confidence sites of phosphorylation. So irrespective of which cutoff we choose, we still see significant proportions of the proteome actually being on non-canonical rather than canonical phosphorylation sites. If we go back then briefly to look at where these come out in the SAX fractions, you can see that most of them, and I'm just going to zoom into this region here, most of them are still coming out as we expected in these later eluting fractions from the UPAC separation. So just to compare then, just to have a look at the database strategy, because obviously we're changing our data analysis strategy quite a bit, going from 3 to 9 variable modifications. If we use the same data search strategy on the same HeLa lysate, but instead separating by titanium dioxide compared to our UPACs, we can see that we do see some non-canonical phosphorylation, but we're talking here now less than 5% compared to over 40% that we see with UPACs. So the identification of these non-canonical phosphorylation sites is not simply a factor of searching for them or looking for them. So there are some caveats with this data, and the rest of the talk is really going to be about the fact that we've now developed the separation technologies. I mean, we've tweaked them a little bit since, but they work, and we know that the phosphorylation of non-canonical proteins, non-canonical phosphorylation of proteins in mammalian cells and human proteins is extensive. The question is, how do we get the most out of the data, and what does it mean? So We have an issue in the fact that, and I alluded to this earlier, that some of our arginine and lysine phosphorylation sites cannot be taken at face value. And the reason for that is that actually when we localize these sites of phosphorylation on arginine and lysine, they actually localize to that C-terminal residue that you get when you digest with trypsin. Now, with everything that we understand about the way that trypsin works, if a residue is phosphorylated on lysine or arginine, it should not be capable of being cleaved with trypsin. So we have to assume that for whatever reason, these sites are wrong. And it can be quite extensive. So looking at these at either 0.75 or 0.99, then we end up between something like between 20% and 60%, 70% of the sites that we identified not being on that C-terminal residue. So there are a number of reasons that we might be observing these. One possible reason, I think it's unlikely, but it's a possibility, is that we have a contaminating protease that can cleave a lysine or arginine phosphorylated side chain. Another possibility is actually that the software that we're using struggles to cytosine using collisional dissociation based fragmentation. So we're actually misassigning that Y1 residue as a phosphorylated lysine or arginine residue. Another possibility is actually we've got phosphosite scrambling, either in the liquid phase or the gas phase. And while I think this is possible, I don't actually think it's accounting for the vast majority of the data that we see. I'm not going to have time to talk about that today, but I'm more than happy to discuss it with anybody afterwards if they want to. Okay, so how do we understand what our false localization rate actually is? So I've been thinking about this now for a few years, and so this actually came out of a conversation that I had with Albert Heck at a meeting in the Netherlands. And it occurred to me that whenever we do biochemical studies, trying to understand the functional effects of phosphorylation, what we always do is we make a phosphonull version, and we mutate a site that is phosphorylated to alanine, because we know alanine cannot be phosphorylated. And then we ask ourselves the question, if that site is then an alanine, it can no longer be phosphorylated, what happens within the biological system that we're investigating? And we can actually do exactly the same from a data analysis point of view. So alanine can't be phosphorylated, but if we search for phosphoalanine, which we know categorically cannot be there, then we can use that phosphoalanine identification rate to estimate what the non-canonical specific false localization rate is. So we did this study, we dropped out phosphocystine and replaced it with phosphoalanine. And then we identified 404 sites of phosphoalanine. And at the time we got this data, I thought, oh, well, you know, the whole thing's not going to work, because 404 sites is more than we saw for almost everything, even histidine and tyrosine. So something didn't look like it was working properly. And then I realized actually that you can't take the numbers at face value. You have to normalize, you have to standardize for these numbers based on the prevalence of individual amino acids. And if you look at the prevalence of alanine versus histidine, tyrosine, for example, there's far more alanine residues in the data, in the peptide spectrum matches, than we ever saw histidine or tyrosine. So of course, the chances are likely increased that you identify these residues as being phosphorylated. So you can use this data to calculate or estimate, I should say, a false localization rate for each individual site of non-canonical phosphorylation. And using these numbers, and these range from 6% here for serine, and then 69% for glutamic acid, you can then estimate, sorry, that was Siri coming on my iPad. You can then estimate the false localization rate of each of the individual non-canonical phosphorylated residues at the different PTMRS values. Okay, so what do we see from the data? This I've already shown you before. So of the 129 phosphohistidine sites, we estimate that we have around 83 true positives, and this is compared to around 99 true positives that we identify using the same approach for phosphotyrosine. And I showed you the consensus before. What I didn't say is that around 57% of the sites that we identify have this consensus, so it's actually really quite a high proportion. We can do exactly the same for phosphoraspartate. The phos localization rate here, as computed using our phosphoalanine search approach, is higher, so it's around 60%. But even with this, we estimate that we have, dependent on which PTMRS value you choose, around 160 true positives. If we take the 0.99 PTMRS score, so this gives us 42 true positives, then we can identify a really nice consensus. It's similar but not identical to that seen with histidine. So we have a proline at +1, a leucine or an isoleucine at +1, and 41% of our sites have this consensus. We can then start looking at what type of proteins contain phosphoraspartate. I'm sorry, I clicked too soon. And the proteins that contain phosphoraspartate tend to be the ones that we see involved in transcriptional control, so poly-A RNA binding, mRNA splicing. So we are hypothesizing at the moment that phosphoraspartate is actually involved in transcription regulation. Phosphoglutamate, the numbers are similar to phosphoraspartate. The degree of high phos localization rate for phosphoglutamate, and this is the highest we see for any of them, means that although we do identify a consensus, I really would not want to draw too many conclusions of this at the moment until we start figuring out how to reduce our phos localization rates. The enrichment, however, of proteins that contain this is similar but not identical to phosphoraspartate. We do see poly-A RNA binding, and again we see a propensity for proteins that also carry another type of modification. Phosphorylation. So phosphorylation, we have lower numbers. We have around 82 true positives, we estimate, with a consensus for either lysine or glutamate pre the site of phosphorylation. One thing that was interesting with both phosphorylation and phosphorarginine is we see this KP-RP position. And actually, we don't think that is necessarily a true consensus for phosphorylation. I think this is probably actually an artifact of the fact that we're using trypsin to generate these peptides, and therefore RP-KP will be in the middle of the peptide sequences that we're looking at. And the numbers for phosphocystine in this particular set of experiments is quite small, so we do have a consensus, but it's based on a small number of cysteines. So what I'd like to spend the last 5 or so minutes talking about is what we think is actually going on with these different types of phosphorylation. And
sorry, Claire, apologies, can you just mind trying muting your microphone off and on? There's a lot of sort of background noise that keeps coming through. And I'm just going to reset the viewer just quickly, just in case.
Okay.
It keeps coming and going, but it's just got quite bad. Let me just reshare it. You'll just have to request control again. Okay, that sounds much better already.
Okay. I've turned down my volume in case it's feedback, but let me know if you... Can you hear me?
Yeah, sounds great. Thank you.
Okay, so protein kinases. So a lot of protein kinases will autophosphorylate. And one of the things we've always wondered is whether or not the protein kinases that we identify as containing sites of non-canonical phosphorylation are actually autophosphorylating. So we went through all the data for protein kinases and actually noticed not only that protein kinases, a number of them do contain sites of non-canonical phosphorylation, but actually there's quite a few that contain sites of non-canonical phosphorylation within the catalytic domain. So an example here is one. So this cysteine residue is phosphorylated, and we've got some really nice, so this is ETHCD data. We can see some C and Z ions and B and Y ions, so we get a really confident site localization for the cysteine residue. But what's particularly interesting is this lies just downstream of an APE motif. And the activation segment of protein kinases sits between a conserved DFG and an APE motif. So this is slap bang in the middle of our activation loop. Now, the reason this is particularly interesting for us is because we've also been working for a couple of years now on redox chemistry and cysteine-based redox. So what I'm showing here is the sequence in the activation loop of a protein kinase called Aurora A, and you can see the DFG motif here, which is responsible for binding magnesium, for magnesium ATP, and here is the AP motif. And this is then mapped onto the 3D structure. And what is particularly interesting is that this site of cysteine is a primary redox site. And when you oxidize this cysteine residue, actually you inhibit catalytic activity of Aurora A. So this threonine residue that lies just upstream of the cysteine is an essential requirement. So phosphorylation of this threonine residue is essential for catalytic activity, but there is interplay between phosphorylation of this residue and the redox status of the cysteine residue in terms of regulating activity. And you can see this here looking at an Aurora A activity assay. So the wild type Aurora A, if you treat with hydrogen peroxide, then you inhibit activity. If you mutate that cysteine residue to an alanine, you don't you abrogate it, you don't abolish it completely, but there is a significant reduction in the effect that hyperoxidation can have on catalytic activity. If we start looking more generally at protein kinase structure, so this is the DFG motif and the APE, as I mentioned before, across all eukaryotic protein kinases, then roughly 12% of eukaryotic kinases actually have a cysteine in that region downstream of this activation site. So this is equivalent to about 100 human serine threonine kinases. And what we've shown is that this appears to be a general mechanism, so that that conserved cysteine predicts redox sensitivity of a whole variety of human kinases. If we link this back to our data now, and these are a couple of examples from the table I showed you before, we have this DFG motif here, and these are the sites of cysteine phosphorylation identified. These are highly conserved, so in this particular protein, that cysteine residue is in 33% of all eukaryotic equivalent enzymes, and both of these cysteines are conserved in 84% of eukaryotic proteins. What's particularly pertinent to these two is actually they have also been identified as being oxidized. So there does appear to be an interplay between cysteine phosphorylation and cysteine oxidation. Continuing then on the idea of interplay, and these are just a few examples. Protein histidine phosphatase, so PHPT1 is a histidine lysine phosphatase. We also observed this to be phosphorylated on an aspartate residue. So the question is then whether or not there is interplay between both non-canonical phosphorylation and other non-canonical phosphorylation. Histones are a classic example of multimodal post-translational modification regulation. And histidine H4 was one of the first proteins in human systems to be shown to be phosphorylated on histidine. And it's well established that the modification code on histones regulates chromatin structure. We've shown that histones are modified on lysine and arginine, and actually one of the sites of phosphorylation, lysine phosphorylation, that we see is also known to be acetylated and ubiquitinated. So there is another body of work that would suggest that actually there is PTM interplay in terms of the modification code of histones. The final couple of minutes, I'd like to really put out a challenge to the community. And this really comes back to this issue about site localization confidence. So a few years ago, we did a benchmarking study where we took a synthetic phosphopeptide library and a large-scale HeLa phosphopeptide enriched digest. And using different fragmentation strategies, so this is HCD using either Orbitrap-based or ion trap-based analysis, and then ETHCD looking at either Andromeda or mascot search engines, and then site localizing with either PTM score, which is integral to Andromeda, or PTMRS. What we did was define cutoffs for site localization that gave us a 1% false localization rate. And for HCD ion trap data, which is what we use to acquire most of the data, looking at STY, then for a 1% false localization rate, we used a PTMRS value of 99.2. When we do our all PX, our non-canonical search, then this is the data that I showed you before, at a PTMRS value, which is roughly the same, then actually, dependent on which site we're looking at, we're talking between a 6 to a 69% false localization rates. And while this is fine to establish non-canonical phosphorylation as a principle that this is important in human biology, it becomes much less useful as we start to unravel the role of individual sites. So the question is, how do we improve site localization confidence? Tom Muir and colleagues published a study a number of years ago looking at whether or not you can use characteristic neutral loss from precursors, either of 80, 98 or 116 for phosphohistidine. And actually we did a comparative study and showed that it doesn't work. And I'm going to run through this data really because I'm running out of time. If we look at HCD, we only see 17% of our spectra contain this triplet neutral loss. So this is loss of all 3 neutral loss ions. STY, we see about the same, between 23 and 26%. And even with our phosphoalanine search, which we know is rubbish, we don't know what it is, but we know it's wrong, we still see 16% of these spectra still contain all 3 neutral loss ions. And it is largely irrespective of what PTMRS score that we have. So the neutral loss, the triplet neutral loss, is not predictive, for example, of phosphohistidine for HCD data. So at that point, we needed to go back to square one. And so we took a whole library of synthetic peptides and our phosphomyoglobin that we generated with potassium phosphoaminate. And we did a multimode fragmentation where we subjected these to HCD, ETHCD, ETCID, CID, or UVPD. Okay, and then we started looking at the mascot scores. These are generally okay, apart from UVPD, fragmentation wasn't very good. And then the PTMRS site probability. And on the face of it, these all look brilliant. But when we actually look at the phosphorylation sites, HCD actually mislocalizes to an aspartate residue at D4. And it gets worse because then both HCD and CID also potentially identify with confident localization scores the glutamic acid at position 2. And the mislocalization that we saw with HCD and with CID appears to occur independent of whether or not we allow these search engines to consider neutral loss from the product ion. So this appears to be a general issue. Just to give you an example of what I mean, I know that you probably can't see the details in these spectra, but I've annotated these fragment ions. So the green ones are B and Y ions, and then there's a grey one here which shows that this is just a neutral loss. And the problem with CID, or actually all collision-induced association strategies, is that the product ion neutral loss that you get is equivalent to having a non-phosphorylated site. So the ones that I see here, which would suggest that that histidine is not phosphorylated, are exactly the same as whether or not we would have a phosphorylated histidine that has lost 80. If we go to an electron-mediated association strategy, however, where we're looking at C and Z ions, which are now in gold, these retain the site of phosphorylation. And so this gives us much greater confidence of where the site actually is. It's very similar to what we see with canonical phosphorylation. Generally speaking, we have higher confidence using electron-mediated dissociation strategies, but because we typically lose neutral loss of 80 rather than 98 from non-canonical residues, the effect is much more pronounced in terms of site localization. The problem with electron-mediated association strategies is they are typically much slower than doing an HCD experiment, so you have to compromise site localization in terms of the number of peptides that you can identify. So the way that we are planning on getting around this, although we haven't actually done the large-scale comparison yet, is to make the most of the fact that actually the predominant ions you see during collision-induced association, so this is resonant-based CID, are actually neutral loss either of phosphoric acid or phosphates, and use that then to trigger either ETHCD or ETCID. So really the challenge for the community now is, both from a fragmentation and from a computational point of view, is how do we improve site localization confidence? So to summarize then, so based on our findings, existing phosphopeptide enrichment strategies, particular titanium dioxide, is generally unsuitable for the analysis of phosphate phosphopeptides containing acid label phosphate groups such as phosphohistidine. But what we have done, and we have shown this now with a number of model systems, and actually quite a few people have now themselves started using our methodology, is that Sachs fractionation at pH 6.8 can permit separation of non-canonical and canonical phosphopeptides to allow them to be identified. While we cannot currently have confidence necessarily in specific sites that are identified as being non-canonical, we can absolutely say that our understanding of phosphorylation mediated signaling in humans is vastly different to we thought about it, 3 or 4 years ago, because it is much more ubiquitous than we ever gave it credit for. And that was really because of the strategies that we as a community were going about thinking about and analysing phosphorylation in human cells. We're hypothesising that there are potential roles for aspartate and glutamate in transcriptional regulation. And actually, we have some quite strong evidence now to show that there is interplay between phosphorylation of cysteine and redox-regulated cysteine that may affect protein catalytic activity. The triplet neutral loss is not indicative of phosphohistidine, but it may be used in a kind of roundabout way to facilitate ETHCD-mediated non-canonical site localization, which may improve throughput. But actually, the big thing is we hypothesize on a lot of roles, but we still don't understand what the regulation of these sites are. So I think this is probably going to be most of the rest of my career trying to figure out what these are. I can see Catherine there. But one final thing I'd like to hypothesize is actually about numbers, and this is completely, hand wavy, back of an envelope calculations. But if you consider the ratios that we have, so this is the true positive predicted values, looking at the ratios of the different non-canonical phosphorylation sites, then based on the current numbers that are in the Phosphosite Plus database that have been identified 5 times or more, then we predict that these are the types of numbers that we are likely to end up seeing from human proteins. And if you think about that numbers, and you think about how I started in terms of the pharmaceutical industry's drive to target protein kinases, then there's an awful lot of scope, really, both in understanding their role in disease and also potentially in targeting their regulation in terms of therapy. So just a recognition of the fact that most of this work has been done by Gemma and Chris. So Gemma left the group a year or so ago now, and that work was picked up by Chris. And funding in particular from BBSRC and Northwest Cancer Research. So thank you, everyone. Sorry for the noise in the middle.
Thanks very much, Claire. I can hear myself. There was some sort of feedback. It was an absolutely fantastic talk, and I think what you've done is to expand the proteome to a number that none of us can now compute very easily. If you think of all those different phosphorylations being combinatorial. So we've had loads of questions, and I don't think we've really got time to do that many of them justice. So Claire, if you wouldn't mind going to the Slack channel and answering any that we don't manage to cover now. But I think one of the ones that got a lot of ticks for people is a purely practical one. This is from Nicholas. Are there specific things to do or avoid during sample preparation? Now, obviously pH is one of them, but are there any other tips that you'd like to share with us if we all want to go off and try this at home, as it were?
Yeah, so it's quite a different strategy that we would use in terms of lysing cells. So ordinarily we would lyse cells in ammonium bicarbonate and Rapidgest because it breaks everything apart. You absolutely can't do that for this because, Rapidgest or any acid label surfactant is going to require acid cleavage. So we do use urea. You obviously then have to be really careful about temperature controlling to avoid carbamylation effects and that kind of thing. Avoid freeze thaws. Don't make the mistake of thinking that you can stick your sample in a fridge for a week and go back to it and analyze it. You can't. The other thing that's probably worth mentioning is when you load these samples onto the reverse phase column to do your LCMS stage, then we load these in water. We do not add acid to the vials for LCMS separation.
That's great. There's very good tips there. Just following on from your last point, do you keep your gradient short on the LCMS to avoid the peptides sort of sat on the column at a higher pH, at a lower pH? Or
No,
does that make any difference?
They're loaded onto the trapping column under what is near neutral conditions, so they're only exposed to formic acid when we actually start doing the elution. We haven't changed the elution gradient, and this was a very specific choice actually, because the rate of hydrolysis once they're bound slows down quite a bit. So we appreciate that we will be getting some hydrolysis on column, but it's nowhere near as much as you would get in solution.
Okay, what I'm going to do now is to try and coalesce a couple of questions, and this is to do with crosstalk. So we've had some questions about crosstalk. For example, do you see evidence of the phosphorylated lysine that has also been shown to be a site of ubiquitination? Do you see phosphorylated cysteine that's also known to be involved in disulfide bridges? So have you sort of seen any of those types of phosphorylation where it would show that there's very clear crosstalk between two competing chemistries for that particular amino acid?
Yeah, so for the lysine, yes, we have seen lysine residues that are both phosphorylated and reported to be ubiquitinated. We have not confirmed that it is ubiquitinated ourselves, but it's in the database and it's an established site. Yes, we have seen that. In terms of disulfide bonding, we haven't looked. We have done some studies, for example, on some of the Aurora A and some of the other protein kinases, but we haven't actually gone back and done a large-scale analysis to see if there's overlap between disulfide bond formation and phosphocysteine. But we have got a couple of examples now where we do have sites of cysteine phosphorylation where they are reversibly oxidized.
Okay. A question here from Harry. Given the prevalence of non-canonical phosphorylation, could many phosphoserine, threonine, tyrosines be misannotated and actually it be an adjacent amino acid that's modified?
Yeah. So I didn't talk about this, but so we've been doing a lot with redoxylene. We've been working on HIF as well, so hypoxia-inducible factor now for a number of years. And actually, we did also identify a site of cysteine phosphorylation on HIF1 alpha. And that was actually a complete fluke because we used titanium dioxide. So cysteine is not a phosphoamidate bond. It is not as labile as histidine. Sorry, we haven't yet done the study to look at half-life under different pH values, but we will be doing that. So we found this site of cysteine phosphorylation using our standard titanium dioxide based enrichment protocol. And I have just become obsessed with looking for everything. So we found it that way and we found it reproducibly. There is no doubt to my mind that that's there because there's no other STYs around it. It couldn't possibly be anything else. But Harry is absolutely right. It has made me now wonder whether whether some of the data that is reported, particularly for something like histidine, which we ourselves have found in a standard titanium dioxide based enrichment protocol, may actually be misannotated.
Okay, and another one, if I may, and I'm paraphrasing actually what the question is. You didn't go into the kinases and phosphatases that are likely to be involved in this system, but are we to assume that, like with the very well studied phosphorylated amino acids, that actually the non-canonical phosphorylations are going to be equally as reversible? So I suppose the question is, are there phosphatases that are going to come along and take these off pretty readily?
So PHPT1 is a known histidine phosphatase in cells. It's also got in vitro lysine phosphatase activity. Yes, I presume that there will be. We don't know what these are. You know, it's a big question. Whether or not some of them, we don't actually know that they're all enzyme regulated at the moment either. We have evidence that some of them may be, but that doesn't necessarily mean that they all are.
Okay. And if I may paraphrase another question. It was just to do with rearrangement of phosphorylation in gas phase post-ionization.
Yeah.
I suppose a manifestation of whether that could be true or not, we'd be looking at your studies where you were seeing phosphorylated C-terminal lysines and arginines.
Yeah.
So for those peptides, was there any evidence of consensus sequences for kinases that were phosphorylating serine, threonine, or tyrosine within those peptides? Did you sort of look for consensus sequence enrichment in peptides that would seem to be containing the C-terminal phospholysines and arginines?
No, actually we haven't. That's a good idea. So in terms of looking for an SPTP motif, that kind of thing?
Yeah.
Yeah, no, we didn't.
Okay, and Harry's just popped up another one. Is there a strategy for heavy labelling phosphorylation?
Well, yes, there is, Harry. That's generally with 32p or 33p, which no one in their right mind would stick near a mass spectrometer.
Okay, and is there a database available for abnormally phosphorylated proteins which cause different diseases in humans? That's a very general question, I suppose.
Abnormally phosphorylated proteins. Well, we are the only ones that have got this amount of data on this many proteins, and we haven't done one, so no, as far as I know.
Okay, that sounds like a grant in the writing, that does to me. I think we should probably leave it there. I hope I've tried to get the essence of the questions. I've tried to combine some of them that have had similar topics. So Claire, if you wouldn't mind having a look at the Slack channel and directly answering anything that I've missed. But I think in the interest of time, we should bring the session to a close. So thanks very much, Claire. Thanks, Ben. I think we've had a fantastic afternoon. It's very hot and steamy outside, so it's very nice to sit and listen to sort of hot topics within proteomics at the same time. So thanks very much. I think my job now is to hand over to Jo. So I will do that now. Thank you.
Great, thanks Catherine, and thanks Claire and Ben.